All right, well, why don't we get started? Thank you all for coming. This is our last CAC Wednesday discussion of this semester, but we already have most of next semester planned, so look out for the schedule. We want, for next semester, we want to welcome especially Dan Kainer from Biotechnology today. Thank you very much at the end of the semester for putting this all together for us, Dan.
My pleasure.
And Dan has brought a panel of student researchers, which he'll introduce, and their title is Project Greenspace, Research Opportunities for LSC-Montgomery students who want a cleaner and greener world.
Thank you. Find mine. All right, so I guess I'll go ahead and start with introducing this illustrious panel. We have students and a professor from Tomball who is helping us with one of our projects. So we have Sean and Tim and Tony and Carmen and Amarani and Yasmin. And they're all going to tell their stories. And I'm hoping that less of me, more of them is a good thing. So I'm going to give the overview as quickly. Hey, and we have another one, a former student who is one of our researchers. This is Michelle Coleman. So we have a lot of stories to tell. I'm going to try to give the big picture. And what we'll do then is let them talk about their individual contributions to this project. And then if you want to ask them questions or anything else, please feel free to do that. So let me get started. I think the most important aspect of a project like this is a good acronym. So Project Greenspace is what we have here. And we do research on campus primarily to be a very novel educational platform for science education, especially STEM education. And we want to teach the students really what scientists do on a daily basis, and that makes sense. And for a lot of historical reasons, our research is centered around two sides of pretty much the same coin, which is clean, renewable energy and environmental remediation. So we ventured off into a little bit of different territory, which we'll also talk about. Besides that, primarily that's where our focus is. All right, so as a biotechnician, we like to think about problems, so that gives me a chance to think about solutions. Some of you may recognize Richard Smalley, Nobel Prize winner, discovered buckyballs. He famously came up with a list of what he found to be the 10 most daunting challenges facing humanity in the next 50 years, and he actually ranked them. And I think just for time's sake, we won't talk about why he ranked them this way, but suffice it to say that his contention was that energy was the primary need of humanity. If you solve energy, pretty much everything on the list is more solvable than if you don't solve energy. So, and when you say energy, it's clean, renewable energy. So energy and water are by far the two most pressing. And then in various orders, this is his order, we could debate this, but there's food, environment, poverty, terrorism and war, disease, education, democracy, and population. I don't think anyone would dispute that these are important problems. We might squabble about the respective order. But what I'm proud to be able to say is that the projects that we have on campus impact most of these. And so let me show you what I think we're doing at least. We certainly are working on energy. We're working on water. We're working on food. We're working on environment. Indirectly, at least, we're working on poverty because all of these things can lift people out of poverty, and we're working with companies directly to do specifically that. We're not really helping with terrorism and war. Of course, if you help with poverty, maybe that helps solve it to some extent. The educational platform, I think we have a fairly revolutionary platform for educating students on the community college level. And even though I don't think we're doing anything about population, I think what we are doing is impacting many things that are impacted directly by population, so some of the effects of population. At least we can indirectly address that one as well. So, what is it, 7 out of 10 of the 10 most important problems in the world? That's not bad. That's all we want to do, solve them all. All right, so let's talk about some other problems from an educational point of view. First of all, opportunities, and we'll see kind of a dichotomy in opportunities and issues. One is there's right now in 2012 7.4 million STEM jobs in the U.S., and for those of you who aren't familiar with that acronym, it's science, technology, engineering, and math. In 2018, they expect that to go up to 8.65 million, so there's a lot of jobs for STEM graduates. Right now, there are 600,000 STEM-related manufacturing jobs that are unfilled. These companies need workers with particular skills, and they're struggling to find them. STEM labor force is growing at twice the rate of the overall labor force, and they make more money on average, even if they don't go into STEM careers. So there's a lot of advantages to being a STEM graduate. Well, with all that being said, you'd think that people would just be lining up and joining STEM programs, but not always. Here's some problems. You survey high school seniors and you see that 17% of them who are proficient in math, because these aren't the ones who are struggling in math, but those proficient in math, 17% are planning on majoring in a STEM field. 16%, let's talk about the formatting, 16% of college grads have a STEM degree. Even those that enter into STEM degrees, here's an article that was just, I believe, last month or September. There's a very significant amount of students who start in STEM and leave it to go to other degrees. And the question is why? We're all struggling with this. So sometimes it's lack of aptitude and interest. Sometimes science is viewed as boring or intimidating. Maybe it's not cool, as those types of things. Maybe there's not enough either qualified or at least exceptional teachers who are motivating students. Maybe it's the way, in some cases, that we teach it, that sometimes there's a confusion about scientific literacy. It's just knowing a whole lot of facts. And yes, it is important to understand the foundations, but thinking like a scientist is a lot more than being able to memorize the enzymes in the Krebs cycle or something like that. STEM class is often perceived as difficult and boring. Well, something that we, I don't want to do that yet. Something that we try to do is show our students what they kind of knew, and all of us who are in STEM, and probably many of you who aren't, know, is that science is fun and interesting and exciting, and kids are born curious. So what I want to do to move into the next part of this is show you a quick little video by a person that I think is doing a very good job of communicating the excitement of science to the masses, both scientists and non-scientists alike. His name is Neil deGrasse Tyson. Some people say that he has kind of taken up the torch of Carl Sagan. And I want to, just this little video that talks about why he thinks scientific literacy is important.
Yeah. I'll answer two parts. One is, what do you do with your kids? And kids need to be able to explore freely. And if you look at most households, they're not designed for that. They're designed to have the kid not explore. The kid comes into your kitchen and pulls out the pots and pans and starts banging them. What's the first thing you do as a parent? Stop that, you're getting the dishes dirty. Yet these are experiments in acoustics. Okay? Whatever the kid is doing, if it has the chance of breaking something, you're going to tell them to not do it. Without thinking that that's the consequence of the experiment that they're conducting. Every time the kid wants to do something, provided it doesn't kill them, it's an experiment. Let it run its course. Even if it makes something messy. You agreed to have a kid in the first place? Fine, clean up after. Because it's those seeds of curiosity that is the foundation of what it is to become a scientist. I don't want everybody to be a scientist. That'd be a boring world. I want poets and I want musicians. And we need that, and I don't have it. But I'm talking about promoting science and literacy. And so the first step for the parents is to get out of the way. Allow the child to explore. If they start playing in the mud, don't do that in the mud. I just cleaned those pants. You're getting in the way of another experiment. They start plucking the petals off the flowers you just bought from the florist. And you say, stop that, I just paid $10 for the flowers. Had you let that continue, they find in the middle the stamen and the pistil, and they learn something about the flower. For $10. That's cheap. Derek Bach, one-time president of Harvard, once said, if you think education is expensive, try the cost of ignorance. The problem is it's not going to boost the economy next quarter. It's got a time horizon longer than most people have the patience for, and most politicians have the re-election cycle to be tolerant of. So what we need is a longer view on those investments. I don't want to have NASA going hand in hand trying to get money to stimulate the frontier of cosmic discovery. And that frontier now involves biologists in the search for life, chemists in understanding the soils of Mars, aerospace engineers. You know what I don't want to do? I don't want to stand in front of 8th graders and say, who wants to be an aerospace engineer? so you can design an airplane that's 15% more fuel efficient than the one your father flew. That's not going to get him. But if I say, who wants to be an engineer and design an airfoil that'll fly in the rarefied atmosphere of Mars, I'm going to get the best students in the class, and you know it, because that's an exciting project for smart people to work on, motivated people to work on. And when you have them, they invent stuff, they discover things, they transform the culture in which we live on a time horizon that is not easy to just tell someone in a one-sentence soundbite. And what I want is a level of science and cultural literacy that will allow the public to be able to think beyond the election cycle, to think for themselves and say, this is a good investment.
Well, in our, you like that, huh? In our small little way, I think that's what we're trying to do here. And I, like in tongue in cheek, I call these guys my kids, but that's what I learned long ago. And I was interested that he used the same phrase I like to use, is that once I learned to get out of their way, that's when the really exciting things started happening. I think you're going to hear about some of the particularly exciting things that our students have been able to accomplish with these research platforms. And actually, you're just going to hear a sampling. I just have a select population. We have many others where these came from. But let me tell you quickly about the themes of research that we're engaged in. And all but one really are environment or energy oriented. So algae to oil, that's the one that's getting a lot of the attention. We're doing lab scale and pilot scale. Algae oil experiments, it's for both biofuels and nutraceuticals. That's a species of algae that we have that produces something called astaxanthin, very, very potent antioxidant. So oil doesn't have to mean what you turn into fuel, but it can. Oil to biodiesel. When we say oil here, our ultimate holy grail type project would be to get lots of algae oil and grow our fuel right on campus where the oil is converted into biodiesel. But in the meantime, we kind of do what the commercialized industry does, and that is use waste vegetable oil and other vegetable oil sources, feedstocks to produce biodiesel, renewable energy. We work on water cleanup. Sometimes, again, that's two sides of the same coin. Algae needs water to grow in, so if you have wastewater or aquifer water, brackish water, that kind of thing, the algae, or other organisms for that matter, can remediate the soil, the water, the environment that it's in, grow nice and plump, and get lipids and for the various projects. So sometimes you can actually clean up the environment while you're producing something important. Microbial fuel cells. The fuel cells are basically machines that generate electricity as long as there's a fuel source. Microbial fuel cells are fuel cells that use organisms, bacteria specifically, to generate electricity. So we're not just into the transportation fuels, we're also into electricity, two main pillars of energy that needs to be solved around the world. Aquaponics, we've been specifically charged, we have a company who funded us very generously to help poor farmers who are struggling in places where there are water crises, where they can't maintain traditional crops. And aquaponics is a fascinating new, not so new really, but a fascinating type of agriculture that we can implement our algae into. But basically the gist of it is that we raise fish, and the fish produce waste that support, that go to bacteria that convert it into useful nitrogen forms, and that supports plant growth. So you're simultaneously raising fish and plants, and you do so in a very, very economical way, in a way that uses a fraction of water, and many advantages over traditional agriculture. We have a solar and wind project. We've always been kind of dissatisfied that a lot of this renewable energy projects have to be, pumps and bubblers and so on have to be plugged in. So we have to use non-renewable sources of energy to create or generate renewable energy. Well, if we had solar, wind, and other types of renewable energy, we don't have that dissatisfaction. So we are actually in the process, and we're working with Tomball right now and the engineering technology program to install a solar array and 2 wind turbines so that much, if not all, of our greenhouse operation, where a lot of this is happening, can be powered with clean renewable energy sources. And then finally, the one that kind of gets off the beaten path is the bioinformatics and supercomputing. And I'm probably the least qualified person in this room to talk about it. Just suffice it to say that modern biology is very good, actually modern science is very good at generating enormous amounts of data. So much data that the old manual ways of sifting through the data just aren't sufficient. So it requires very sophisticated hardware and software to mine the data to draw conclusions that would be missed really by a manual looking it over. And we're going to talk about some specifics there, and then have a couple demonstrations for you. So that's I'm more than enough. What we need to do is let me release this to the panel, and we'll hear about some specifics where the real excitement actually is. Who would like to go first? Okay, go.
My name is Tony Baum, professor at Tomball campus, and I'm professor of engineering technology. And what we did was, Danny and I found an opportunity for a small grant. And what we did was we purchased some solar panel systems and 2 wind turbines. And basically what we're going to do is we're going to create our own electricity, and we're going to power up the greenhouse for him, where he's going to grow the algae to harvest either for biodiesel or whatever. What we've done is the students, or my students that are in the electrician program, as well as the solar photovoltaic, which is solar panels, and wind turbine students, are actually meeting up here. We've been meeting up here a couple of Saturdays now. And later on this month, we're actually physically going to start installing these systems for Danny's greenhouse. Yeah, there's one of the solar panels. There's 24 of those. Those are 200-watt panels. And we have 2 small wind turbines that we're going to put on 27-foot towers. And we're going to put it right behind the greenhouse.
And we do have the opportunity to have this totally off-grid, so it can be run, if we want to, without having to plug anything in the grid generator.
And then we have a set of batteries for backup power.
And his students have been coming every Saturday,
Mm-hmm.
actually since May. We took a little break in the summer.
Right.
But they planned this entire thing out, and
We did have a little schematic that the students came up with.
this was not me, trust me. I would have killed someone by now if I tried to do this. But this was their students that came up with the schematics.
On the left-hand side of the 2 turbines, and I said, like I said, they're on 27-foot towers. And then on the bottom right, there's the solar array with 24 200-watt panels. And we're going to provide 120 volt AC power at 60 hertz. So he can do anything he wants with that power.
You will, trust me. All right, great. So I thought what we do is everyone gives their few minutes, and then we'll open it up to Q&A for the audience as a whole, if you'd like to. Okay, you want to go next? Okay.
Ben Line. My name's Tim Hall. I'm involved in 3 projects. One was biodiesel, and I'll start with that one real quick. Basically, we was doing converting waste vegetable oil with primarily with methanol, but I switched over to ethanol. And Dr. Kainer passed around a couple of our things from coconut oil and sesame seed. That's actually kind of got a sweet smell to the coconut, and sesame's got a pretty pleasant smell. And when we burned biodiesel that we made from some waste vegetable oil, we did have the scent of French fries coming from the exhaust. I was focusing on the ethanol because methanol is a toxin, and we had a loss of glycerol from the methanol. We couldn't process trying to separate the two. With ethanol, it's non-toxic, it can kind of evaporate off, and I started a side project talking with Dr. Harless about trying to use thanatrophs that feed on methanol as a pure sole carbon source, and I was able to find one. Haven't identified it yet, but able to find one that was surviving on about 9% methanol, which is kind of high. So I incorporated part of the biodiesel research, just trying to find different ways to produce a clean byproducts that can be used. Glycerol can be used in lotions and soaps and stuff like that. The microbial fuel cell, the one that this one?
Oh yeah. This was my prototype fuel cell. When I got involved with the fuel cells, it was under Dr. Kainer. I was already involved in the biodiesel. And Dr. Kainer needed to get some fuel cells up and running as quick as possible. And I just finished Dr. Harless's general microbiology course, and the way that they was doing just didn't make sense to me.
And normally the process was about five days to 2 weeks before you get a reaction of energy production. And I asked Dr. Kainer if he would mind if I approached it differently, and based on an idea I had. And so I tried the first one, and I had a reaction within 24 hours versus 5 days to 2 weeks. And then I told him I had one more idea. And when I did that, the power was immediate, and it was doubled what they were producing already in the lab. Normally these cells operate, we had papers there, ecstatic if it lasts for 2 weeks, 3 weeks. This one right here has been going for 3 years, and almost 3 years, and I haven't done nothing to it, so I'm just kind of monitoring it. So I decided that was the prototype, and I kind of found out by accident. I put these as points just to test the soil, but I get through with my test, and I just had a what if kind of thing, and I found out that at different levels the soil is producing different levels of voltage and milliamps. So I designed the second one because the byproduct, one of the byproducts is water, so I decided to put a drainage tube, and I got, with these points right here, I can measure throughout the cell and have it consistent. I've been able to graph data of what's going on. And through that, I found out through sterilizing some mud and dirt that there's voltaic action there, a possibility there, but the microbes is what are actually generating the current. So that's one of my research projects right there. And part of it has to do with what you see up here that got me involved with algae that I tried to stay away from.
You held out a long time.
I tried.
Wrong one.
Are you going away from the fuel cell?
Well, I was trying to.
I did want to mention one other thing about a student who's not here right now.
One of the problems that I noticed with algae, and this is how it got me involved, I needed something to, basically I wanted to use, algae has a source of enhancing the oxygen concentration. The top of the tube has a magnet to kind of pull the hydrogen up to help the circuit. And algae was having a lot of problems with keeping it growing and crashing. So I decided I wanted to try an experiment, and I got algae growing, and that's how I started getting involved. I used the algae basically from these PBRs to create a water seal, so that I got a concentrated enhanced oxygen on top of the fuel cell. And this right here is where I'm being able to test a different strata that I was telling you about. But this is what got me started in the algae. And then here recently there was a startup company was asking me about, well, this is what the algae is investments, but what I was getting at, I'm trying to hurry because I only had a few minutes. They was at a test meeting, I decided to test it against mine. And the results are right here. I've been doing different concentrations. This is the media that they have been using. It's kind of yellowing, not very happy. This one right here is the media I've been using. They were both inoculated at the same time, but they started growing a full 3 days faster in this medium. And where this is already crashing out, this is still healthy and still going. So I've doing some variations of tests on concentrations of that to see just how far it'll go. And then another sidebar that happened this past week is one of the concentrations is causing it to autoflocculate. One of the problems that is having problems is harvesting. And right here, usually have to run it through a paper, and it's kind of a film. But when it autoflocculates, this is a very small sample, but it pulls all the cells out of suspension. I can siphon off the water and pick it up like this as a paste, and which is much easier, and it's still green and healthy, where that's kind of brown and nasty. This is still green and healthy, and as it dries out. But basically, it would make harvesting much easier if we can repeat this. And I think with the notes that I've taken, I believe I might be able to repeat it. So basically, I tried as fast as I could. That's what I got.
I'd like to add one other thing about the fuel cell project, just because it was such a. And we have another student who's not here right now. She graduated and is in MD Anderson now. But Mary Hughley, she took a different approach to the fuel cell. The thing about Tim's approach is that he, instead of trying to engineer a fuel cell, he took a functional, intact ecosystem and is basically trying to characterize it. We still don't know exactly what organisms are responsible for the fuel cell that's outperforming almost every fuel cell we see in the literature right now, and by the way, very well-funded labs. We're not one of those. But what Mary Hubley did,
Sorry to interrupt.
Mary Hubley, who's right there in the background, she actually took a molecular genetic approach to try to, and she didn't take it so far to apply it to Tim's work, but she used a technique called real-time polymerase chain reaction, which is a very ubiquitous technique in biotech, and she amplified some housekeeping genes, some highly conserved genes, to try to develop an assay that would allow us to identify organisms without having a predetermined idea of what we're looking for, which is pretty powerful. Sometimes your biases, your hypotheses can, well, bias what your answers look like they are. You can go in blind and go fishing for particular organisms from a genetic approach. I just wanted to mention that because when we heard her give her presentation about her project, I really thought I was listening to a grad student talk. It was just so sophisticated and so well done. And she was one of these students, and there's so many stories like this, but she was a student, very shy, very quiet, very reserved, and she just, well, Tim is this way. Oh, so many of these students are this way. They just blossom when they're giving ownership over some of these projects, and it becomes their own. And Michelle Coleman back there, I think she can say a few words to that regard too. She's made many talks like this for us. So anyway, I just wanted to let you know about Mary as well, even though she's not here with us today. Okay, who's next?
I'll go.
Okay.
I actually haven't got a whole lot to say. I'm a computer science major, and I recently found out, actually I asked Professor Iyer if there was anything that I could do related to some work that I'd done before I returned to school, where I had been doing a little bit of work with informatics and artificial life, which sounds like biology, but it's not really. I mean, it's kind of inspired by some principles that we have learned in biology. But in any case, she pointed me to the corner lab where Professor Johnson was, and I found out that he had this cool exotic hardware, like this is a Tesla. It's basically a really, really powerful graphics card. How much does this cost? Like, $4,000, something like that.
The top end, probably down to about 25. So.
But anyway, it's really cool. Like, normally I wouldn't have access to something like this. And he's also putting together a cluster where that's you have a bunch of computers working together on a problem. And so one interesting thing I've learned is that the general theory used in bioinformatics is the same as the theory that I had seen in the informatics that I was using on the other project. So that's cool for me to learn. Like Dr. Stankotter just told me yesterday that there's informatics within physics, like in cosmology. So that's really cool to find out. So it's generally useful for the student body to work on a project like this. And hopefully what's going to happen is that we're going to start building up an infrastructure and get a project going that the computer science students are involved with. It'll have this momentum, and it'll keep going from semester to semester. So hopefully we'll be able to establish that. Really starting in the spring is when I'm going to get heavily involved. At this point, I've started writing some software. I'm able to compute a couple simple bioinformatic metrics for 6 million base pair sequence. Download it into a graphics card. And do it. It takes.
Just no big deal. No big deal. Little project.
Every time.
But it.
Are you the one that fixed about the web page?
But I mean, the reality is that our current state of knowledge in computer science is behind where our hardware is. Traditionally, our advances in computing power have come in processors that become faster and faster in doing things in series, and we're reaching the limits of that. And now we're moving into this parallel processing. And with our current state of technology in programming languages and the way that we as computer scientists think about writing code, it's extremely difficult to implement the algorithms that do it. So I don't know what to say beyond that. It's really hard. I'm learning a lot. It's a great experience being able to work on this sort of hardware. I mean, I'm actually extending my stay at Lone Star by a semester so that I can do this. Because I don't expect that I will be able to transfer to a four-year university and immediately start working with faculty to do informatics research on what amounts to a supercomputer. I mean, we've got, it's really not an exaggeration to call this hardware a supercomputer, because it is the same sort of setup, maybe not at the same scale, but it is the same sort of technology, maybe with fewer nodes. So it's a fantastic opportunity. I've already got 2 students working with me on the project. That I have, I worked in industry for over a decade as a programmer, so I already know what I'm doing. Not in this particular domain, but programming generally. But I've got 2 students who are getting involved. I hosted a talk last night for the computer club, and after that, more students came up to me and they said they want to get involved with this. So I think that I think great things are going to come out of this. So.
And I have to say, so the presentation last night was Skype. Where was that from? Where was he?
San Francisco.
Larry
Yeager.
Yeager, 20-year software engineer with Google, and there were a lot of questions about what does it take to get into Google. And they were asking, can we do it with a bachelor's degree? Can we do it with a master's degree? Can we do it without a degree if we know what we're doing? And I'm going to toot your own horn there, but Larry Yeager, 20 years at Google, pointed to Sean here and said he's one of the most brilliant programmers he's ever known. And we have the privilege of having you working on our project. So thank you for that very much. All right, who's next? Want to keep going around the table? All right.
Well, good afternoon. My name is Emerani Morales. I've been working on aquaponics, and I became interested after I attended Murray Helm's workshop.
At the beginning of the semester.
Yeah. He was talking about it all through summer, so I was like, okay, I'll go. So what captured my attention was that it's self-sustainable, and you can grow 2 cash crops, plants, and fish if you know how the system works. So after the workshop, I talked to Dr. Kainer that I wanted to start a little system before we started the big one. Yeah.
Couldn't wait for us.
So, I was like, okay, I'll make a little one, try to learn as much as I can, and then learn from my mistakes and come and apply everything I learned to the one we have here. So I had no idea how to grow fish or plants, and unfortunately I killed 47 fish. 49.
We learned by failing.
Thank you, Edison.
And I didn't know how to grow seeds either, but after a lot of research, and
That's
I
great.
tried a lot of different ways how to grow them. So I found a way to grow the seeds, and like 90 to 100% of all the seeds I planted germinated. So I was really happy. I wasn't expecting that. So even though they germinated, I killed more than half because I would pull them too hard or something. So I was able to grow these. These are pumpkin seeds. And on this picture, it's kind of like a hydroponic system because it didn't have any fish nutrients, kind of. And then my dad helped me build the other one, all the aquaponic system. So he even helped me build a greenhouse. So
Oh, I didn't show that picture, sorry.
I was really lucky to find this lady who was giving away 16 greenhouse panels. So it saved me a lot of money. But that's like the greenhouse inside of the greenhouse, and that's the upper thing. The upper tub is the grow bed. Yes, that one. That's where the plants grow, and then the lower tub is the fish tank. So, and then on this one, I transferred the pumpkin plant to the aquaponics system, and it's growing really big, and it looks strong, so I'm really happy.
You brought us a sample
Yes,
for us.
this is kind of like a hydroponic system. So I planted 2 lettuce and a cilantro. and they're growing.
If you're not familiar with the difference, hydroponics is where the plants don't, both aquaponics and hydroponics, they don't grow in soil, they grow in water. But in hydroponics, it's a dense nutrient solution, whereas in aquaponics, the fish waste support the plant growth, so it's more close. They're related, but not exactly the same.
So
What type of fish did you use
I use catfish.
Okay.
Yeah.
Like baby?
They're like 6 inches right now.
Where do you get them at?
There's this man who comes from Oklahoma. He sells them for 69 cents each. That's maybe why I bought
That's
it.
I can understand that. We also have several, you want to talk about Green Life and your experience there so far?
Yes. So I've been volunteering at the Green Life Hydroponics Company, and they've been teaching us how to take the pH of the fish tank. They're telling us what the nitrate and nitrite levels have to be for the water. They're teaching us how to plant seeds, and what else?
You're doing the fall harvest, or the fall planting.
Yes. So we're learning a lot.
They're getting some labor and for some free training in return. So. Let's keep going around. We have so many people here. David, you want to talk a little bit about?
I haven't done much.
Come on.
I'm an all-around person.
He's an all-arounder, but he's always there. He does a lot of little things. Just
Yeah.
give it a couple highlights.
I-
Very humble.
Just various different projects. I'm just kind of spread myself way too thin.
Yeah, he was there all Saturday, this past Saturday, prepping the greenhouse for a lot of work. We got pretty dirty.
Huh?
All right, Michelle. Michelle is a graduate of our program, and I'd like her to mention a minute about the project she did here and what it led to for her as a vocationally, where she is now.
I am Michelle Coleman, and I'm a graduate of the Biotech program here at Huntsman. And I first heard during a little workshop get-together that Dr. Kainer was having about algae. I think it was during our bioinformatics class. I can't remember. I think it's the first time I heard it, or molecular. I can't remember. And it just kind of spurred my interest. And I said, well, what can I do? And I'd like to volunteer. And I started volunteering with the big photobioreactor that we had down in the greenhouse at one point. And they were 12-inch tubes, about 5 foot high, and there was 6 of them. And so I just kind of took over the project and volunteered on a daily basis. And kept doing it, and finally earned my internship and started running. We were transitioning to the new research or project lab, and I helped start that up and gave me a lot of experience. And in algae, also doing water remediation.
Remember this thing?
Yeah. So,
Here she is in the Chronicle.
So I graduated and I found my focus, but where to find a job? Well, I was very, very lucky. One of a professor who no longer here, Dr. Larry Loomis Price, was doing his sabbatical at Huntsman Advanced Technology Center. And the supervisor he was working under needed a chemical technician. Was not me. Chemistry was not my strong suit. And but who had a background in algae research. And so he volunteered me and I got the job. Basically, I got the job because I knew algae, not because of my chemistry skills.
On the job training.
Yeah, on the job training with that.
Lots of chemists over
So-
there.
Really, not lots of, yeah.
Exactly. So, even though he keeps pushing it. But that's what kind of, it just, I was in the biotech program, I was floating, what do I want to be, what do I want to do? It's so versatile, and this gave me a focus and gave me a drive of what I wanted to do, what I wanted to work with. And algae is so versatile in itself. You can do so many things with it that we're already putting it in cosmetics and personal care products. We're trying to get enough feedstock to actually make surfactants to put into our personal care products and detergents.
One of the things Michelle dabbled in just a little bit before she left us for literally greener pastures, right? is, she worked on a water remediation project. And I'd like to mention that for a second because that one is something that was made possible because of a very generous donation by a company called Aparian. And Aparian is a property development company in New Mexico. Well, they're based in Arizona, but they're working on a big project in New Mexico outside of Albuquerque. And New Mexico is a very severe water crisis right now. And so that not only impacts the people, but it also impacts industry. Intel has a big plant where they're trying to develop this property. And so Intel was threatening to leave, and Intel, I believe, is the second or third biggest employer in New Mexico next to the state government. And that was going to hurt a very impoverished state even more. So this company went searching. This was like a decade's search, long before we got involved, for water. And if you can't get it from above, maybe you can get it from below. So they tapped and managed to finagle the mineral rights to a very large, very deep fossil aquifer. And the problem is, and it's about 5,000 feet under the ground, 80 miles wide, 40 miles long, something like that. But this is what comes out of that aquifer. And what they're trying to do is they're trying to build what they claim to be the world's first completely sustainable master plan community. Something akin to the woodlands, but based on sustainability always. And sustainability means you produce as much or more than you use, right? And they're going to do this on a large scale. We're talking a $100 million project and so on. And they saw that we were working on algae, so they asked us if we could, instead of working on algae production, if they could work on algae remediation. So this is a sample of the water they want us to figure out how to clean up. We haven't had much luck with it because it's horrible. It makes the water in Galveston look pristine. Actually, they decided that maybe they should have started with something like that, just to not be so challenging. It turns out that things have changed. They gave us a $82,000 grant. Not even a grant. I didn't have to write it. But they just gave it to me and let me do what I wanted with it for the most part. It was pretty awesome. And we the next thing that happened is they changed their focus and they decided not to wait for this technology to emerge. And they said, we're going to go with traditional water remediation. What we have is a much more acute need. We have farmers who are losing their farms because there's no water and they're very poor. Can you use algae to help them somehow? And that's actually where the aquaponics came from. They gave us another grant. This one wasn't quite as big. This was about $31,000. They put them together over, six-figure funding from this company, and it was sort of, it was really philanthropic. The CEO of the company, there he is. He's talking to Dr. Lane there. He makes Howard Hughes seem tame, but some of you have met him. Some of you have been frightened by him. Dr. Lane didn't know what to think about him. But anyway, his name is David Maniotis, but he, as hard as he can be to work with, he's very generous, and he really is very passionate about turning this whole science literacy thing around, getting students excited about science. And if we can have research platforms that, first of all, give them a better education, but at the same time, maybe we can get a few discoveries or technological advances that act as proof of concept that they can implement on much larger scales, well, that's all the better. That's sort of the icing. The education is the cake. And he really has signed the checks and backed off and just gave us general directions. And what he wants us to work on is water remediation in the first place and some sort of agricultural solution, which we've chosen aquaponics to be the most reasonable as a second. So, okay. Jamie.
Hey, I'm Jamie Upadhyay. I was a computer scientist until I came here to get a biology credit to continue computer science at U of H. And then I saw a biotech flyer saying free pizza. I think it's one of Dr. Kainer's Get Volunteers Quick scheme. It worked. And so I went to it and we started talking about everything. I thought we were philosophers. We were talking about nanotech and biotech and the future, basically. So I asked him if I could do research, and of course, I was in the research lab a week later, and I couldn't touch enough things there. I first started on the biomass project. We were just focusing on trying to get as much algae as we can produce, and so he told me to start working on one algae was Chlorella vulgaris, And so I thought this banana-shaped algae was Chlorella vulgaris for the longest time. And then Tim pointed out, this is something else, something we've never seen before. And we realized it was an algae strain called Scinadesimus, and it was taking over everything in the lab. And so I'm sure we're still having problems with that, at least in another facility. But after that, I moved on to bioremediation. Professor Johnson had gone to the Galveston Bay Area and collected different sources of algae. And one of them was from, well, most of them, actually they were all saltwater algae except for one that was from a biorefinery. And so the main goal was to clean that nasty thing up. And so none of the salt water, that is more or less salt water in between, brackish. But none of the saltwater algae did well with that for some reason, we don't know. But the freshwater biorefinery algae, over time, began to clean it up, and the conductivity went down because we think there was high metals in there. And so we were seeing progress there. But we never really sent it off to see what was in there, so we couldn't really further our experiments.
So working on increasing the functionality of our lab. Little by little.
No, but I just wanted to do everything I could possible. The first day I signed up for everything that we did, everything. And then I didn't know what I got into the next week.
It's like a steamroller, right?
Something. But another thing Professor Johnson was preaching was the algae called E-Hux, or Emiliana huxley. And this thing apparently could do so many things. It has 2 genomes, apparently. I don't know much about, ask Professor Johnson about that. But there's literally country-sized blooms in the ocean of these. And I have a picture of here I'll pass around in a second. The blue is actually the bloom of the E-Hux, and that's the coastline, obviously. And so the main aim, or the algae we got from the Galveston Bay Area, some of them actually, Professor Johnson thought would have E-Hux in them. So he told me to go look for this thing, do it with really no experience. So we did lots of research on the web, and all we could find was electron microscope images. And that was nothing to go off of, because we had a light microscope. So
We have an electron microscope, it just doesn't work.
yeah, but we actually think we did discover some. They look like gold coins, if you know. But once the semester started, I couldn't really take care of it, so they all died. And that's why we need more volunteers in there.
And I have new water.
Pardon?
I have more water.
Oh, you do? Okay, there's more water on the way. But yeah, so they, I think the last cell that I found died a week ago. And then, so I moved to another project. And this one was the most productive, or one of the most productive ones, and it was the Hematococcus pluvialis algae is what I focused on. And this one was known to produce astaxanthin, an antioxidant. You can see it by that blood red color over there. And so, but the other difficulty was trying to grow it because it's so sensitive to its environment. And for example, we had a tank in the lab, and it was labeled Hematococcus pluvialis, but no one had bothered to actually check what was in there. So this kid who was taking a summer class, who had never been here before, just said, okay, well, let's see what's going on over here. And found out there's these banana-looking algae. And Tim's like, dude, it's not happening. So that was the Scinadesimus, and it was just taking over everything. So luckily we had Hematococcus pluvialis growing in the greenhouse. And so I took some of that, started growing it, and me and a couple of interns decided to stress it. But we had to make sure it was on its own in its environment, because other algae might be eating up taking all the nutrients that would die. So we had it isolated, and we had a control grown it was grown with basic nutrient solution. And it would have like 12 hours of day, 12 hours of darkness. We had 2 other ones, which was pure darkness, and one was pure intense light. And they all had salt in them. And a few weeks later, we found out that they actually started turning orange, the ones in pure, intense light. And so, but we weren't able to get it blood red like you see over there. And I don't think many people in the world even know how to do that. So,
Struggle.
yeah, no one really knows exactly how to do it. But as far as I got, I made it look like an orange, basically. And I have some pictures here of pre-stress and post-stress. A picture of E-Hux that we think we found, and then a picture of the balloon in the sea, and a picture of the E-Hux in the electron microscope. Want to pass that around?
So you said did you change your major?
Yes. Oh yeah, now I'm doing biotechnology associates here. I plan on going to MD Anderson to get molecular genetics and possibly going to medical school for neurology. But, that's all over there up in the air. So.
We got him away from you over there.
Yeah, I did actually. Wait, that's about it.
Pretty good.
You really want to talk about your experiences quickly with, yeah.
Well,
The
for
high
me,
points.
it's recreation. Just a chance to work with students, like Tim, that take things on. I've also used work in the either in the project lab or in the greenhouse with my non-majors biology class. When we had them writing a couple of papers during the semester, they were tired of writing papers by the end of the semester. All their other classes had write papers, and so I would give them the option of doing time in the greenhouse or the project lab instead of writing a paper, and then they could do their presentation on. So they didn't really have time or the background to actually do experiments, but they would help other people with their experiments. Or I think over a couple of semesters they were instrumental in getting the little greenhouse erected.
Yes, that was a job. I've never.
And so part of it was, that it was tended to be people who were really good with their hands and like doing physical work, but then they had to do their presentation on why were we building a greenhouse. So we got a lot of people convinced that algae is going to save the world. So they learned some science, but they could also see that not every scientist is somebody in a white coat and a lab bench with a micropipetter. That we need people that know how to do things, like these guys, and that there's an outlet for doing all kinds of stuff there. So they really seemed to enjoy it and got quite enthusiastic. This semester I had 2 students in the majors class go to the aquaponics lecture. One guy, he was showing me pictures. Instead of pictures of his children on his phone, he has pictures of his aquaponics setup and how he's done it. So he's thinking of our aquaponics setup as a place to try things before he does it for real in his. But I'm thinking we've got some expertise to tap into here because he's ahead of us. And so he's gotten much more enthusiastic in the class, the non-majors biology class, because this ties in somehow. He's not sure how, but since I'm doing it, talking about it and suggesting this, and I'm teaching biology, they must connect. So he's doing aquaponics, but doesn't quite understand how it connects with biology. Okay, so I think we can tap in and use these people's interest you didn't know that you had, so even if they don't end up changing their major, they at least are feeling empowered about biology, and that they can do science because it's something that they're already doing, or it's something that they're interested in, and they just didn't know that was science.
Want to hit a couple highlights, Ricky?
Why don't we let these
Okay.
two go?
Yeah.
They want.
Sure. Just going around.
I-
So we need to turn this off, or?
Well, do you need it?
Okay. I'm not very good with technology.
It's not bio.
It's bacteriophage.
Oh,
Well,
oh, you need the shield.
Okay.
Okay, we need this down first or up first?
I do that down
Yeah.
first.
We might do it again.
So, my name is Yasmin Ansari, and Carmen and I are working on the two different projects in bioinformatics. What got me interested in bioinformatics was for one of the classes for special topics, Dr. Kainer took us to the different research labs, and the supercomputers really caught my attention because the two supercomputers together work as much as 1,000 computers, close to 1,000 computers. And also what got me interested into bioinformatics was that there is so much data out there, and there's not enough people to look at all the data, and that we could actually make discoveries by just going through all the data that people have collected over all these years and make discoveries. So the disease that I'm studying right now is alveolar capillary dysplasia with misalignment of pulmonary veins. It's ACDMPV for short. It basically affects the development of lungs and the blood vessels around the alveolar sacs. The lungs are basically like an upside down tree and they keep branching, so it affects the exchange of oxygen, and it affects newborns because the development happens before we are born. So the infants suffer from cyanosis right after birth from a few minutes to a few hours, and it's extremely fatal. So so the gene that I'm focusing on is the FOXF1 gene, and it's actually a regulatory gene. It controls other genes, and its protein controls other genes, how the other genes make their proteins. So it has the piece of DNA which I'm focusing on within the FOXF1 gene is the intron, and this intron is an interfering intron. It interferes the other RNA from other genes. So I'm just going through the collection of genes that's online on the NCBI website. I'm doing something called blast searches, where I put in the sequence of the intron, and it goes through all the genes that are available and finds matches. So I do this online, and then after that, I'm going to start using more of the bioinformatics applications. For this, since I got so interested in bioinformatics, I actually changed my laptop and my PC's operating system from Windows to Linux, because I find Linux to be working much better now. So that's something that is really cool. I'm trying to learn Linux more and more.
So we need this off.
I need it on for the first part.
Okay. Did you download it?
No, I was just gonna open it. Let's go away, but.
There it is.
So my name is Carmen, and the project that I am working on is comparing staph genomes. And for you guys who don't know, Staphylococcus aureus, it's a bacteria capable of causing very serious illnesses in humans, and some strains of Staphylococcus aureus have been found to be resistant to antibiotics. And there are many different strains with slightly different genomes, and currently there are over 33 strains genomes that have been sequenced. And so on classifying genomes, how it was done kind of before was you would kind of look at the characteristics of the organism, and you would break them down into different categories. And so, you know, the example is like, you know, group A has all black hair, group B has all brown hair. But now it's going on to more of a microscopic level, so looking at their DNA specifically and looking for the similarities and differences, and using these similarities and differences to create meaningful categories. So now it's more like group A has a gene that codes for antibiotic resistance, and group B doesn't. And then so specifically what I've been working on is I've been looking at two genomes. It's called USA 300 strain and the Newman strain. And what I'm doing is I am taking the two genomes that I pulled off the internet, and I am creating a dot plot, and I'll pull up the supercomputer and show you all this in a moment. I'm creating this dot plot using the program called Eugene, and I'm looking for areas where they are different. And for my particular project, it happens where I have to use a supercomputer, because we tried comparing the two genomes on just your regular computer, and it would not load. We thought we broke the computer. But then we went on supercomputer. It took it a few seconds, but it did. It took a few seconds, but it did it. And then so after I identify all these chunks, you know, these chunks, some are unique to the particular strain, so it's not in any of the other strains, but some of them are similar across multiple strains. And then, so I'm gonna take these chunks of DNA and I'm gonna compare it across the other 10 strains to kind of be able to classify them into distinct categories with very distinguishing chunks. So like, for example, strain 1, 5, and 6, they all belong to group A because it all shares 1 or 2 chunks of DNA that are similar. And then from these similar chunks, we can complete a search to find out what these chunks of DNA code for and give these categories meaning, not just, you know, they all belong here because they all look alike. It's like, what does this group who looks alike is actually going to do? So now I'm going to show you the software.
All right, so we need to turn off the projector, I guess. Right?
Yes.
I don't know how to do that.
Danny, if you just make the slideshow again and hit D, it'll turn it black at least.
That's Google.
Yeah, that's right.
Google, so I don't know.
Okay, so hit B?
Yeah.
Oh, I didn't know.
I didn't know that either.
Technology, right?
Yes, good.
That presentation
Hit
right
B.
there.
Hit B.
B means blank.
Gotta like it. Not control.
No, just B.
Nice. Wow.
We better lights out.
You know where I heard that?
That's good.
It was like 2 years ago. There was a presentation by a guy from Rice University, before the school starts. And it was it's about how to make a good PowerPoint presentation. And it's like, oh, hit B. That makes where your class will stop paying attention to PowerPoint. Start paying attention to look at it. I don't know the time.
Cool.
Yeah.
Look at how she's got way too many of my students in here for this.
You do your tricks.
See, it's taking a few, it's running its progress bar. It wouldn't run a progress bar on the regular computer. And so this is the dot plot that it generates. It's plotted currently right now the Newman strains genome versus the USA 300 genome, which each have close to about 3 million base pairs of DNA. And what you see where the straight lines are, that's the areas where they have similar DNA, and then these big gaps here, and then you'll see, you don't see it really well on this, like if you click on it, you know, and you click zoom, it'll actually zoom it in for you, and you can actually see, you know, kind of where the differences in the DNA is, and then adjust this, and then you can kind of go down here, and it'll actually give you the actual DNA sequence that it kind of corresponds to. So that
So you can click on the line on your
Yes.
graph, and then it tells you the sequence under.
yes, it's the box part corresponds to the line, and then it'll give it to you for both strains of DNA. So like the top one here is Newman, where it corresponds to. The bottom one is the USA 300, where it corresponds to. That's my work with the supercomputer lab. That is this far.
Thank you. Did I miss anyone going around the table? And we have Jana Houston over here just itching to get started. She's a first semester teacher, so we're giving her a break. All right, well, if anyone would like to ask any of the speakers questions about something technical or non-technical.
I have a question that's legal. And you all are doing science, but you're also doing real world applications. Does there come a point in any of these projects where you wonder, do I need some legal protection on this process? And then the other question would be, are you at the point in any of these where you really want to consider getting this out into the bona fide professional literature if you're not already doing that?
Well, I passed around, it's not any of the data, but we have been publishing the professional literature this month, or last month now, it's October. So this has been recognized by the Journal of Industrial Biotechnology, the work that we're doing here. So that's, and I've presented this work in all over the place, all many different states. So there's a lot of people who want to do what we're doing, and we've been just, some of the circumstances I described with industry collaboration has really made it possible with non-traditional funding sources and things like that. But as far as the intellectual property, I think is what you're, Tim, are you interested in intellectual property at all?
I've actually, on the fuel cell, there was a possibility of looking into patenting some of my test vessels, and some of the reactions and some of the findings that I'm getting, we're considering publishing on that, so I'm doing some work. Right now, with the setup that you've seen, I have so many because I'm doing 2 sets of repeatable, you gotta do them 3 times. So I'm trying to do as many tests as I can repeat before publishing some of the work that I've got on the fuel cells. Recently, about what I'm doing with algae, there's a startup company that is kind of looking at me right now and wanting to possibly help me patent the feed that I've come up with and license it for me and then go work for them. So I'm getting involved in an area that I am having to consider this in a way that I haven't thought of before. So I'm still treading lightly to find out just what all it's in. But yes, as I've told some of the other students, don't underestimate what you can do in that research lab. Some of the stuff you can get to do, you may could be significant, and just give it a shot and try.
This Tim presented preliminary work at the microbiology meeting, so there's an abstract out with this stuff. But then we started kind of backing off on publishing writing up the whole paper, because maybe it was patentable, maybe not. And if you publish it, then it's not, once it's in the literature. So, but then I think we found that some of the stuff was probably not as unique as we thought.
Well, the test vessel is what you see up there.
Right, but the data, we might be able to write it up without giving away just all of the secrets.
Right.
So we're working.
I came across some, I found out that the U.S. Navy is doing something kind of similar to what I'm doing. I'm in a different, complete structure what they're doing, but their approach is the same. So there's some of that I just really don't know the ins and outs of, which requires legal.
Maybe it's a little more funding than Tim has, but you
Well,
duplicated
one of the
their-
problems
their
is that if you
efforts.
work at a university, they're gonna have a technology transfer office and then they will patent it for you and work for it. Our system doesn't have that. And
Except, one thing that did happen because of what Tim was doing is that it came to the attention of the chancellor. And he saw this as a very unique branding opportunities and really into branding Lone Star as premier community college, all that. And he basically said, and apparently there's been board policy that has been created specifically for this reason, that if a student develops intellectual property, he didn't say faculty, but if a student develops intellectual property and Lone Star sees it as appropriate, Lone Star will help secure the intellectual property, and all the rights for that IP will go to the student. And the reason he wants to do that is because it brands Lone Star as a unique place where you can come and make discoveries or develop enhanced technologies and have the opportunity to secure the intellectual property. And so the hope is we can attract more Tims by offering that possibility. So.
have they gotten on this?
Don't know.
So we don't really have an office and a mechanism like they would in a big research university, that they would have a whole office dedicated to looking at, has anybody got anything patentable this week?
Well, we didn't pursue it because when we found the Navy was doing something similar, we kind of backed off pursuing it. But
maybe for
we'll put it to
vessel?
the test at some point. At some point. But. Any other questions?
To the point of besides patents, thinking about publishing, Yasmin's research on Tox F1 was initiated by someone at Baylor College of Medicine, and with the intent that someday this would be published. So in that sense, yes, we're interested.
So it's an opportunity. We're discussing. We're in talks with MD Anderson right now. They have a molecular genetics technology program. Jamie mentioned he wants to go to that, and they have a requirement that their students do research rotations. So they've already initiated, and they came to us when they saw what we were doing. But they want to talk about sending biotech students, because all our biotech students are required to do internships. And those students who want to continue on and continue their education at MD Anderson maybe could do their internship at MD Anderson, start their research project even before they begin their MD Anderson classes, and then kind of have a head start. We have 320 internship hour requirement, so it kind of gives them a 320 hour head start on a research project that could lead to publication and or presentation at national conferences as well. Oh, and by the way, they partner with Yale and Emory and Duke and all kinds, and send students to those places as well. Harvard Medical School. I've heard they're doing pretty well.
So everything that MD Anderson does isn't necessarily explicitly cancer research related?
99.9% of it. So when we talk about internships, now, the focus of this talk was on what we do on campus, but not all this is an option for our students, not a requirement. So a lot of our students, they're just laser focused on getting into the workforce, so we send them to industry, usually, to do their internships, and they just get industry experience doing all kinds of different things that have nothing to do with, sometimes they do, right? But oftentimes have nothing to do with what we discussed here today. And that MD Anderson would be an example of that. They would be involved in cancer research. I think I was told that MD Anderson has 10,000 scientific publications a year, and 99.9% of them are on cancer. So chances are, yeah, they'll probably be on cancer, most likely.
You must have quite a list of alumni in graduate schools and so forth.
We could do 3 more hours of this if we wanted to. There's so much happening. It's thrilling to be part of it. I think Julie will tell you, Vicki will tell you. We see levels of student engagement that just are off the charts. I mean, Tim, we can't get rid of Tim. He graduated. he won't leave.
But then it's really stimulating for us too, because the students come to us with questions that I don't know the answer to. All I can help with is an approach. And it's like, this is what I was trained to do, is figure out how to figure out something. And so I then have to go, I was at MD Anderson as a graduate student. We didn't do algae there. I learned a lot because they needed to know. And I'm good with the search engine. And kind of feed them information and we can talk about approaches, but we work with them like they were scientists because they are, and they've got questions and we're trying to answer questions. And send out our colleagues.
Well,
which
there's
is
no
really
question
cool.
about it. You all are, students, you are bona fide scientists, there's no question about it.
I alluded to it at the beginning when I was rushing through the overview that it's a platform. It's an educational platform, and what I didn't say is that the traditional platform in kind of, we call it cookbook chemistry, no offense to chemists or anything, that there's a result that they're supposed to get. The instructor up here on the stage, if you will, knows what that result is supposed to be, and it's supposed to turn yellow, but oh no, it's turning green. What did I do wrong? And so they're trying to figure out from the professor how to fix it. That's good training because it's troubleshooting. But this, the professor has no better an idea what the answer is.
I don't know if yellow or green is the right answer.
We discover it together, and that's what scientists do. So that's not the same exact. It's just a different, more realistic training platform that gives students an insight into what scientists do and experience on a daily basis.
I know that I'm sorry, I just want to add this real quick because it adds on it. I know it accentuated my critical thinking skills. It from coming where I was so based on biology and then coming to a chemistry lab and thinking of algae in a whole new different light and what I could do with it, I'm able to critically think that now. I'm able to take this piece and this piece and try to put them together to figure out a solution. Where before, it would be mainly on the biology side of things.
Well, I must say that there is an analog in my own education. The greatest professor I had in grad school, Professor of Medieval Literature, and what he would do is we'd sit around and he would say, all right, let's puzzle, help me figure this out. You know, rather than, well, did you read my book? You know? and it was authentic. We were trying to figure out, we were trying to interpret it as a poem as a group. And he didn't come in with an agenda. And it's kind of the parallel to what you were doing as scientists. You know?
And I bet he was surprised
Yeah,
at some of the insights that you guys.
Yeah, and it was the greatest experience I had in school.
And it's almost unheard of for freshmen and sophomore students to have experience. What these guys learned to do, I mean, that's what I did for my master's degree, right? Is learn how to work in a lab and ask questions. And they're gonna be walking in to graduate programs.
Hit the ground running.
Yeah,
Yes.
they're gonna get out in 3 years with a PhD, you know, it's like, okay. Whatever.
Cheaters.
Because you know how to do science. I had to learn how to do that in graduate school because I knew how to read a book and remember the facts.
Right.
The director of the Molecular Genetics Technology program, MD Anderson, asked me if I could fill his entire class with Lone Star Biotech graduates. I said I didn't have enough. So.
Just something I wanted to add too from the student's perspective, not to talk to Dr. House and Dr. Kainer both about it. When I started working in the lab, the classroom was just trying to pass the course.
Exactly.
And box
Well, no,
science.
it wasn't.
Just
It
trying
was,
to
no, it was just making the A do.
but whatever, to learn it, and then we was trying to do the right mechanism to get the right answer. But when I went into lab and started doing my own research, now I see why the Krebs cycle is so important, the electron transport chains, and then when I started the physics class, I wound up getting 2 degrees instead of 1 because I was taking more courses to understand what I was finding. And so the class.
Ever in college to take an extra math course so we could take physics, so we could understand it.
So, and it was just, I wanted to understand, and the whole perspective of the class changed. It was like, can we hurry up and get to this chapter? Because I really want to learn this, I want to understand this, and it changes the whole dynamic of how you approach the class. Once, just from doing that over there, you see the relevance of what's being taught.
Exactly. I totally agree.
You all are scientists, you can figure out a way to clone, yeah, so that we can all have it in our classes.
That's the next project.
Absolutely. Well, thank you, Bobby.
Very well.
Well, everyone, please help yourself to cookies and cake, and thank you all for coming. And thank you all for your presentation.
Thanks for having us.
I had to come by your office because
Weekend on well.
did it help you that? I want to show you the apparatus. I have a very hard to show you how it works. But actually, I don't show you so an appointment time. But just to let you know. So this is the thing where they challenge me. I'm like, okay, well I get it. Like I know how